Hello, and thank you for joining us today for the roundtable discussion and part two of the GastroPlus user group webinar, Utilization of PBBM PBPK models for building a safe space and regulatory applications in support of drug product quality. A few housekeeping notes before we get started. For optimal video and audio connection, we recommend you close out any additional web or streaming applications that may affect your internet bandwidth. We take your privacy rights seriously, and by attending this event and participating in the Q&A session, you're allowing the GastroPlus user group committee to contact you for further follow-up. This webinar is being recorded for future playback on the Simulations Plus website and YouTube channel. You'll be able to find part one on the YouTube channel, the LinkedIn GastroPlus user group page, and the Simulations Plus website to review. And as always, if you need assistance throughout today's webinar, please raise your hand or use the chat function. On behalf of today's panel, we appreciate you spending your time with us. And now, before I hand things over to Tycho, a quick poll question. Are you a member of the LinkedIn GastroPlus user group page? We'll give you just a few seconds to answer. Thank you everyone for responding. If you are not yet a group, go ahead and choose the last option and we'll be sure to get you the link to join the group where you can find more peer discussions and find out how your peers are currently using GastroPlus. All right, without further ado, I'll turn it over to Tycho.
Thank you, Arlene, for the introduction. Today's session will be more conversational and more interactive as compared to the part one of the webinar. Today, we are happy to have several expert panelists from 4 different companies and also from Simulations Plus. We have Sandra Suarez-Sharp, Vice President of Regulatory Affairs at Simulations Plus. Vera Lukakova, who serves as Chief Scientist at Simulations Plus. My colleague Sumit Basu, who's Senior Scientist in the Quantitative Pharmacology and Pharmacometrics group at Merck. We have Talia Flanagan, who serves as Head of Biopharmaceutics at UCB. And David Sperry, who is a Senior Research Advisor at Eli Lilly. And we also have Christoph Testert, who's Principal Scientist in Biopharmaceutics at Janssen. We will go through approximately 20 questions. These questions have been collected based on user group feedbacks from the first session and also hallway discussions. You can think of this event today as a help desk question session or a GastroPlus clinic. You can think of it as a roundtable, if you will. And prior to going to the questions, I will pass it on to Sandra Suarez, who had given a webinar on September 8th, and she will take about 3 to 5 minutes with a recap of that session.
Thank you, Tycho. What I'm gonna do is just show my screen here. See, okay. Everybody can see my screen?
Yes, we can, Sandra.
Great. So again, good morning, good evening, everyone. And I wanna thank you one more time for joining us today. Before, as Tycho mentioned, before we can start with the discussion, I'd like to give a very brief overview of the topic I presented back in September 8, which is the use of PBBM model for establishing a safe space and what are the regulatory applications in support of drug product quality. And I'm not gonna launch the presenter's view because I'm gonna be flipping over the slides.
I cannot hear you, Sandra, if you're talking.
Oh my goodness. You should have told me. I've been talking for, okay, so I don't know what happened. So let me go back. I think I apologize for that. I don't know what happened here. Maybe I pushed the wrong button. Anyway, start again. So I wanted to give an overview of my presentation, and one key thing that I mentioned is that the pharmaceutical companies are seeing this drug product development and life cycle management as a burden, as seen in this survey. So they do want to implement changes, but I see this as a burden due to the complexity around the regulatory framework. And so I mentioned that there are many ways to diminish this burden. And one way to go about it, or to solve this problem, is to establish a safe space. I define what was safe space, which I want to make sure that we're all on the same page. I want to repeat it. Are the boundaries defined by dissolution profiles within variants within this space are anticipated to be bioequivalent. So means then that safe space actually is the same as bioequivalent safe space. If you were to take a look at a publication that came up from a workshop that we had back in 2017, the MCRC workshop that was organized by the FDA, we have a definition there of safe space, and also we call it bioequivalent safe space. And so those two are the same. However, safe space is not the same as knowledge space, or may not, all depends, right? Just to revise, what's knowledge space? The knowledge space is the set of data, or in this case, observed in vitro and corresponding in vivo data used in building the safe space. Let's say you use 3 formulation variants that were tested in vitro and in vivo. That constitutes a knowledge space. If you were to use, and within that, you can define what's the boundary of releases that are bioequivalent, right? If you only use 2 of them, let's say the upper and the lower bound, to define your safe space, and they happen to be bioequivalent, and in that case, safe space is the same as knowledge space. Also, not to complicate things, we must not confuse the concept of safe space with the concept of design space within the product quality world, right? Design space, I would say, is a function or a relationship between all the critical process parameters and material attributes. And it will be the same space. Design space can be the same, equal to safe space. I would say only has been verified, for example, using biopredictive dissolution specifications, meaning dissolution and acceptance criteria. So recapping, safe space is the same as bioequivalent safe space, is the same as dissolution safe space, but may not be the same as knowledge space, and may not be the same as design space.
Thank you. And if I can ask a question to the industry representatives, you can see, based on Sandra's slide here, that there is a non-BE curve included. So that suggests that non-bioequivalent data or clinical data is a beauty for setting up the knowledge space. I was curious, among the industry expert, how often does it occur that you have non-BE data in your safe space analysis or in your PBBM work?
It's Talia here. I can make a start on this, and then maybe my other industry colleagues can comment. This is one of the really interesting points of debate, I think, around development of a safe space, and also around development of PBBM models to support dissolution specifications. Because on one hand, having a non-bioequivalent batch really gives you a high degree of confidence that your method and your model could detect and reject other non-bioequivalent batches. On the other hand, if you have a well-behaved API and a really robust formulation, it should be incredibly hard to make a batch that is not bioequivalent. And you can end up in a situation where, in order to make a non-BE batch, you would have to make some really significant changes to the process and the formulation that then maybe mean, you know, your dissolution mechanism isn't so relevant to your actual clinical formulation because you've had to change it so much. So I don't think there's a straightforward answer to this one. I think non-BE batches are extremely useful, but if you have to expand, you know, if you have to go to manufacturing and formulation conditions that are really not that relevant to your actual clinical formulation, then maybe that batch becomes less useful. I guess also you would have data from development about formulation changes that you've made, so you would already be understanding the impact of the likelihood of changes that you make impacting on BE. But for me, this is really an important point of discussion. I don't know what my other industry colleagues feel about that.
This is David Sperry. I would say, based on my experience, first of all, I agree with everything you just said. Based on my experience, I would say that it is not all that common to actually have a non-BE batch available for a given development program. And so, as Talia said, it's extremely useful, but it's not always data that we have, either because it's extremely hard to get, like the case that Talia said, or it just doesn't fit into a particular development program.
This is Christoph, and yeah, I would agree with that. Every program that looks for setting safe space, clinically relevant dissolution spec, they're almost craving to have that non-BE batch. Sometimes it's just, yeah, the relevance of your drug product that you're testing can be questioned, and how much do you have to change your actual product to get that non-BE batch? And also in the end, when you get your non-BE batch, we always have the question, what is an actual non-BE batch that is useful? These dissolution profiles that you look at here are still quite close. But if it's completely off and you're at 10 or 15% bioavailability of your target drug product, is it still a relevant non-BE batch to set your specifications or using PBBM modeling? And how about the interpolation within that zone between what you know is bioequivalent and then the broad space to what is non-bioequivalent?
Yeah, so I would just want to add that we do from regulatory polite was at the FDA, we understood that challenge, and want to say that that didn't preclude precisely the approval of the submissions. One thing, that, of course, that we need to understand is that there is not confidence on the model. One thing is, if you don't have that non-BE batch and you have generated data, you already know that your batches somehow are bioequivalent, right? So you could establish a safe space via conventional approaches. And so I would say that PBBM really helps to somehow, as I mentioned before, define that in vitro in vivo link and define that safe space. But because regulators don't have confidence on extrapolating, of course, depends case by case, then the only limitation is that safe space, that the dimension of the safe space is okay. It is not as wide as one the companies would want to have, right? But so again, one beauty of it is that you have the data, the solution profiles with different release rate that happen to be somehow bioequivalent, then you can establish a model so that the advantage of it is, even you cannot expand it, is at least that you know that in that area you can use it to support any manufacturing changes, as long as the model predicts that is within that safe space. But again, the bottom line is that you can still build a safe space with non-bioequivalent data, and FDA, in my experience, has approved models that did not use non-bioequivalent data. And
Thank
of course, to expand that, to also to approve those submissions, my experience, FDA relying on additional information to increase the confidence of the application, or whatever regulatory question it was being answered by the model.
Thank you, everyone. So then we can ask the next question, which is simple. When we talk about safe space or biowaiver, do we usually refer to oral solid dosage forms Are we limited to oral solid dosage forms, or would this the principle also apply for, like, say, parenterals?
Yeah, I can start again, Tycho, just because I guess with regulatory experience, I might, yeah, I have that information. I want to start saying that, as mentioned before, Safe Space is a tool that could be used as a surrogate for bioequivalent studies and thus support biowaivers. Safe Space, as mentioned before, relies on the likelihood for developing in vitro in vivo relationships or in vitro in vivo correlations. And this is possible with other routes of administration, right? So then Safe Space is possible too. Hopefully that answers the question without being so convoluted. Again,
Yes.
Okay.
Thank you. So question number 3, for scientists who are not so familiar with GastroPlus, can Simulations Plus post a best practice safe space example with virtual trial work case examples that includes the DSD file, IVIVC files, CRD files, STL files, and the MDB files, including detailed instructions which would complement the videos?
So I guess this is a question for me. This is actually something, it's not just about the file, but it is really about the process of how to go through the example, how to go through the instructions, or how to establish the safe space. And it is something that we do teach as part of our workshops. So those of you who have attended the workshop, especially the one on pharmaceutical development, we do have an example linking the in vitro dissolution to in vivo dissolution, showing the example of evaluating the dissolution safe space, what range of in vitro dissolution profiles would still lead to the bioequivalent formulations and so on. For others, I would encourage to keep an eye on those types of workshops, and the files and the whole process is part of that as well. Again, posting just the files would not be helpful. It's really about the process and learning how to set it up in the program so that you can work it with your own examples and with your own formulations. As for the PSA and the output plots, that will really depend on the type of formulation that you are working with, right? What is the main determining factor for the dissolution? Those would be the parameters that you would be exploring for the sensitivity analysis.
Thank you. Question number 4, which % CV for dissolution data are considered too high for PBBM? This relates, of course, to F2 similarity tests where there are some specifications around % CV. So what can you say about the % CV that is too high for physiologically based biopharmaceutics modeling?
I can start this one. I love this question. So one complaint that my analytical colleagues sometimes have about me is they come to me with one question on dissolution, and rather than an answer, I send them away with 5 more questions. And I think that's how we answer this one as well. It really depends what the variability is caused by and what the method is that you've seen the variability in. So if you're working with, say, a biorelevant dissolution method, and you have a high CV because your formulation is genuinely showing really variable performance in that method, then I think irrespective of the % CV, you try and understand the impact of that variability in performance using the PBBM model. On the other hand, if the variability, if the high % CV is caused by some kind of method artifact, some kind of hydrodynamics, then maybe that tells you that perhaps that method isn't the most relevant one for you to be using, or perhaps you could augment it with some orthogonal data in alternative, disso setups, that kind of thing. So this question really made me think, but I think for me it would depend on the reason for the variability. That would be my answer rather than giving a set percentage. I don't know what others think.
Yeah, I can also add into that. Thank you, Talia. I agree with you. And thinking, yeah, I'm not sure if there is a cut-off value for which we can say PBBM will be robust or not. As Talia said, I think what we need to do is to have a clear picture of you know, we need to have a clear picture, because both conventional and physiologically based models that are developed for drug quality applications, for example, IVIVCs, rely on mean dissolution data. So that's why we don't have a clear understanding, because usually when we do this modeling, we rely on mean dissolution data. Also, that determination or conclusion might depend on whether the method is under or over discriminating, as Talia was mentioning. As you know, the source of variability in dissolution data come from either wide process controls, wide specifications, or from the analytical method. So an observation, a critical observation that I want to make here is that before embarking in the development of PBBM or any model, one should make sure that the sources of variability are understood and are minimized and justified. So in a nutshell, to have a reliable interpretation on the model outputs, one should develop dissolution methods with the lowest percent CV at any time point. And the clinical impact of the level of variability should be determined a priori by creating, for example, we can create virtual dissolution profiles and build in variability around the mean, and determine what's the in vivo impact. So that way we would know if it varies that much, what's the impact.
Thank you. And now comes another big question. What are PBBM model recommendations for capsule to tablet formulation switches? Sandra, I'll start with you. Is PBBM recommended for these type of formulation switches?
So let me start by saying that safe space, any way it's built, via conventional or not conventional approaches or mechanistic approaches, is derived from the concept, I mentioned this before, the concept of building a relation on a correlation, meaning that it should be built around the same dosage form and considering the failure modes that are specific for that drug product. That's how we build IVIVCs, right? We don't build IVIVCs between a immediate release and extended release, so we share release profiles within a capsule on a tablet. So therefore, safe space that is built for a tablet cannot be used to make inferences on the quality of another dosage forms. I guess that during drug product development, PBPK or virtual bead trials should be considered for when we can switch that formulation, right? But we can do that early in drug product development to say, okay, I'm now want to switch from tablet to capsule, and I'm going to use PBPK, PBBM to determine the likelihood for my formulation that I'm designing, whether it's going to meet the requirements or not.
Do we have any opposing views from anyone from the industry?
I think it can still have valuable information if a capsule or a tablet formulation that may not be in scope anymore is that non-BE batch that you're looking for.
Right.
I won't say it's easy, but I won't rule it out. It contains useful information as long as you understand what the mechanism is that's different between the release of a capsule, the release of a tablet, and how you link it to, for example, in vitro dissolution data with the opening time that you need for your capsules, having a delay in your profile, etc. These are things that can be taken into account somehow. And if that is your non-BE batch, I would still like to make use of it.
Yeah, this is David. I would agree with that. It depends on the mechanism and your level of understanding of the mechanism, just like you said.
Mm-hmm. And so we are thinking that a tablet and a capsule have the same mechanism of release?
I think
Would
it's
you build
possible
an
that they would
IVIVC?
in certain scenarios, yeah.
That's not necessarily the case, but if you have information or supporting data that it is, or if you have a way to capture the differences in mechanism and still relate it somehow.
So you
I'm
are
not saying
saying
that you have to include it in your safe space, that's something else, but if it is that that part of the information that you're missing, yeah.
Right. So I think we are on the same page that when we are building a PBPK model, a baseline model, or a PBBM baseline model, you can definitely rely on that information to say, my model is able to predict that. But to build a safe space using data from capsule and tablet to define your safe space, and then to say that let's say you have used the tablet and the capsule has a much lower release rate, right? And so you said, oh, these two are bioequivalent in vivo, so that my safe space is going to be defined there. I would be, if I were the regulator, I would be asking whether a release for a tablet, a release profile of a tablet that is as low as what tablet, as the capsule shown, are still bioequivalent. So if that information is presented, then I might consider, we should consider, right? But I recognize that data from tablet and capsule can be leveraged to determine the robustness of the model.
Thank you. I see this topic as a potential separate webinar topic where perhaps one of the industry members can present such a case if they exist. I think we're about halfway through the session, and Arlene, we have another poll question.
Thank you, we do. What percentage of IVIVCs, IVIVRs that you may have submitted to regulatory authorities were based on mechanistic absorption models? We'll give you just a few seconds to answer while we get ready to continue with our questions. All right, just a few more seconds here. And let's see the responses. Okay, Tycho, back to you.
Yes, and here we come with another big question. GastroPlus is widely used in the industry for drug development. However, there are few submissions to the agency using GastroPlus to conduct IVIVC models. What are the possible reasons, and what can we do to minimize this gap? Sandra, I'll let you take the first shot at it. I cannot hear you if you are muted.
Yes, yeah, thank you, Tycho. I'm gonna share this slide here, right? So I presented even this morning, and I mentioned that this database that was created by the FDA from 2008 to 2019, only 3 IVIVCs were submitted. There were PBBM models that had built-in based on IVIVC. And I put here attempted because I don't think they were approved. So the reasons I see why this low rate of submission, first, the question is submission, right? Another issue is approval. But so for the low submission rate, I would say there are several issues, and I'd like also invite my panelist members here to answer that. But I would say that low submission rate could mean that although there was an attempt into building that IVIVC, that the IVIVC did not meet the criteria for validation, and thus therefore it was not submitted, right? Simply because they didn't achieve that. And so based on my experience, one main reason for not meeting the criteria for validation is lack of biopredictive dissolution methods to establish that correlation. And so then one way to obviously to go about it is to develop that methods that are biopredictive early in drug product development, and guided by the use of PBBM. Also, if one cannot really develop that biopredictive method early on, then we should generate dissolution data in different media, including biorelevant, and then test the likelihood for building a correlation. Another very important question that I want to raise here, and I also highlighted as part of my future directions on my presentation here is whether the acceptance criteria should be expanded. So I'm wondering whether the current criteria that is established in the guidance, which is no more than the percent prediction error should be no more than 15% for the individual, and for the mean should be no more than 10% even for external validation. I'm wondering whether it is too narrow for this kind of approach. So we need to think about that. Another quick reason perhaps is why they are not submitted is because companies are not aware of this. They don't have the knowledge and understanding on how to build the relationship, so, or perhaps they have not generated the minimum data that is needed to build the model, right? So I would say that would be some reasons why we don't see companies building mechanistic IVIVCs. I like to invite my colleagues.
Yes, I'd like to hear from the industry representatives. What can you recommend other companies to encourage IVIVC, or what can they do based on your knowledge to minimize this gap?
Or maybe also turning that question around, if you are not submitting it, what is preventing you? What is your own experience as to why you are not using it more?
So this is David. I could answer a more general question. I mean, the question here is about IVIVC, but I would say you could generalize it to say regulatory arguments in general. And in that case, what we're always trying to do is find the simplest argument that suffices for the point we're trying to make. And I think that PBBM models are still perceived as being somewhat complicated for making a regulatory argument. And that might be the reason that they're not used in IVIVCs or other regulatory arguments. And then what can we do about that? I mean, I think that PBBM models are not in principle, not that complicated, and could be used more readily in regulatory arguments. And there's perhaps a level of acceptance that an enhanced level of acceptance that we still need to see to get comfort with that across the industry and regulatory agencies.
Anyone else?
This is Sumit. I would like to add one thing. Like, I think it also depends on the steps, like what kind of question we are addressing with that particular model. So if we can address question by using numerical one or the conventional IVIVC, sometimes there is no requirement to do the PBPM. And since it's a approval stage and we are submitting a lot of submissions, sometimes we often ignore to develop that PBBM model. So it's very case by case. And as you know, as David pointed out, this model is very complicated. We are putting physiological parameters plus mechanistic understanding plus our clinical data. So often we couldn't finish it by time. But later we actually share our experiences in the community.
If I can bring maybe a European perspective, there was a survey carried out as part of the Albedo project. So this was a huge European biopharmaceutics consortium that contained academia, regulators and industry. And we did a survey that we published in 2016, asking the European industry colleagues how often and for what purposes they were using biorelevant dissolution and PBBM modeling during development, and how often they were submitting that to regulators. And we also asked them what they perceived as the barriers to submitting these models in order that we could understand the discrepancy between how often it's used in development versus how often it's submitted. And the top two barriers that they cited related to IVIVC and PBBM modeling were lack of alignment in vision and approach across regulatory regions. So the idea that what's acceptable to one regulatory agency may not be acceptable to others in the context of a global submission. Or I would say perhaps it's more that some agencies have really been great at coming out and saying, exactly where they stand on PBBM, whereas with others it's more of an unknown. The other issue that was highlighted was lack of regulatory policy publications and lack of a regulatory framework for how to use these more modern biopharmaceutics tools in the regulatory context. And I guess that creates uncertainty, which means that even if the biopharmaceutics scientist understands exactly how that tool fits with the overall submission, it can be difficult to convince other colleagues in-house to share this data with an agency. So we had an example which Sandra knows well from her past life, where we had to get a direct question from FDA in order to be able to submit the biopharmaceutics model, in order to say, okay, look, FDA want this because they've asked us for it by name. But even then, we still had quite a significant amount of pushback internally. Do you really need to share all these files? Do they really need all of the modeling files, not just the summary report and all this? So I think the lack of framework and lack of alignment are certainly also perceived as barriers to the wider utilization of these models in a regulatory context.
Yes, very powerful. And this highlights that ultimately we should probably have a PBBM guidance from the FDA, because as you mentioned, Talia, it's difficult sometimes to get line functions and project teams convinced that such model should be submitted to health authorities. I think you also...
I would say a global guidance, right? Maybe ICH or at least guidance from regulatory agencies, so that hopefully the commonalities and differences are noted, and there is an effort for global harmonization, as Talia mentioned. It's a great point that she made.
Thank you. And then the question number 7 we have, it's a little bit more specific, and might have been touched partially on. If clinical non-BE data do not exist and the data cannot be generated due to technical difficulties in making such non-BE tablets, can PBBM be used to generate a virtual non-BE dissolution profile?
No, I can start again, Taiko. I mentioned this before. Short answer is yes. PBBM could be used to generate a virtual BE profile and therefore define the age of failure, right? And this approach is common during drug product development, as I mentioned before, for formulation selection. However, the regulatory application might be limited for, I would say, higher risk situations. And but we could use this approach, as I mentioned in my presentation, let's say for some situations where the risk is low. For example, BCS class 1 compounds that did not meet the criteria for rapidly dissolving. Let's say now dissolve within 45 minutes, and of course, for which the excipients have not changed. I would say that if they are properly validated, there is no extrapolation being proposed, that the rate of success for establishing safe space is high even without a non-BE.
Great, thank you. And here comes another big question. How can PBBM be used to select and identify biopredictive dissolution profiles? I think this is a recurring topic. I don't know that we have a silver bullet answer here, but I think this is a very important question. Anyone? How do you establish biopredictive dissolution profiles using PBBM?
This is David. I can tell you.
Okay.
Oh, go ahead, Christoph. Go ahead.
Okay. Just want to take the first stab at it. I mean, it can serve you as a guidance of what is of relevance, yes or no, and how your discriminatory properties for your CQAs, etc., would be. Especially in early development, it can be very powerful to explore your space and to link it back to your criticality analysis of your drug product, ultimately linking back to your dissolution method. So it's a combination of having profiles available, looking at what is of relevance in your criticality analysis, and applying that with, for example, what we just talked about with that virtual non-BE dissolution profile, it can be a powerful tool to support what is a biopredictive dissolution method.
Mm-hmm. Yeah, this is David. One of the ways that we use PBBM, this is not the only way, of course, to help with biopredictive dissolution is essentially through a mechanistic deconvolution of what the in vivo dissolution profile must be. So biopredictive dissolution methods, aiding in developing those is having the information of what the in vivo dissolution profile is. And you can, of course, with PK data, you could use a PBBM model to essentially back calculate what that in vivo dissolution profile is. And that might be very helpful in developing a biopredictive dissolution method.
Thank you. And now comes a technical question that has to do with GastroPlus dissolution models. Looking at the built-in dissolution models, there are several options, such as the Johnson model, Z-factor, etc. They are basically similar equations, but with different parameter setting. Is there a preference for each model regarding different formulations?
Okay. So this is where maybe I start with this one, explaining what was our thinking in incorporating all of these models. And then maybe some of our industry participants might want to fill in as to what is their common practice within their companies. It is correct that both models, Johnson, Z-factor, Weng-Flanagan, they are all based on the same strategy, on very similar equations. The decision there is based on the type of inputs that you may have available. Johnson, Weng-Flanagan models do require particle size or particle size distribution for the compound. Z-factor lumps all of the parameters of density, particle size, diffusion coefficient into that single value of the Z parameter, which would be obtained from the in vitro dissolution data. Right? So in order to parameterize the Z-factor model, typically you would be starting from in vitro dissolution data. Right? So that is already the first thing to look into, what type of information you have available, which of those models you actually can parameterize. The additional decisions would also come from what is driving the dissolution rate. Right? Is it really mainly the API solubility and particle size? Right? In which case the Johnson or Weng-Flanagan model would be appropriate. Right? Or is there a significant contribution of the excipients? Right? Are the solubilizers, wetting agents, other excipients that you are adding to the formulation really changing the dissolution rate, so it's not driven anymore so much by the API particle size, but more by the excipient. Right? And in that case, you should really go for the Z-factor model, because that one would lump all of those processes, including the excipient effects. So those would be the initial strategies, decisions that you need to look at. But again, if there are any of our industry participants that have additional comments to that.
Anyone?
Yeah, this is Sumit. So I can add like a couple of points. So like suppose when we are doing for different formulation, we also check the compound character, like whether it's a BCS class 1 or 3. So in that case, like maybe direct input is meaningful. We don't have to worry about the physiological one. But for BCS class 2 and 4, since it's a permeability limited, we might have to think about more like additional experimentation or other assumptions. And just like one part of caution, because I have seen in some of the cases that first we get the clinical data and then retrospectively we get the dissolution profile. But we need to think about that our input function or dissolution data might be a range of the data, right? So without any assumption and others, a long range of dissolution profiles would result the similar PK data. So we shouldn't do that without any validation.
Thank you. And here comes another question that has to do with the Weibull functions. Can you describe when Weibull functions should be used in GastroPlus, and if a sponsor submits a Weibull function as part of a regulatory response, how can the sponsor best justify the use of a Weibull function? Sandra, maybe you can answer the question about the justification of the Weibull function, and then Vera can talk about when we should use it.
How about you start first, Vera?
Okay, so I'll actually start from the decision on what you are using the Weibull function for, right? You are typically looking at two profiles. One is in vitro, one is the in vivo profile. And for the deconvolution of the in vivo dissolution profile, right, we do recommend to use the Weibull function because you are just trying to find the dissolution profile that would lead to the outcome that you measured in clinic, right? That would result in your observed PK. And in those terms, it really shouldn't matter what you are using to describe that profile. It's all about having the function that is flexible enough and does have enough functionality to be able to describe that profile in case that it is a more complex profile, right? That you have some burst effects followed by slower release and things like that. And the Weibull function does allow that, right? So it does have that flexibility with fewer optimized parameters. So that is why we chose it and why we recommend it for deconvolution of the in vivo dissolution profiles. When it comes to in vitro profiles, you do have option to either use directly the tabulated data that you measured in the lab, right? Or use Weibull function to smooth it out. And this is where it becomes a question of what is the more realistic behavior of the formulation, right? And it becomes a question more in cases where you have only few measured points, right, for the in vitro dissolution data. If you do simple linear interpolation between measured data points, this can create a profile where at certain time point, right, the release rate suddenly changes, right? So is it more realistic to expect that that is really happening, or that the transition through the release or dissolution rate is more described by the Weibull function? And that will smooth out those transitions, right? And the only answer here is the only way to know which one is closer would be to actually measure profile with more output points, right? Instead of measuring or capturing the dissolution data or dissolution profile with 4 points, right? If you get a more dense profile, you'll have that shape captured more accurately, and the decision will be much easier.
Thank you. Sandra, how does a sponsor recommend or justify the use of the Weibull function?
Thinking about it, because so I want let me give you an example that I experienced when I was at the FDA. This was for an immediate release formulation, and they wanted to determine whether these two different profiles were bioequivalent. And so the way they did it is to fit the in vitro, for an immediate release, fit the dissolution using the whole platform, and they came out with the factors that define the shape, right? And so they say, okay, because these factors that define the shape, the critical values that come up from the Weibull function, because they are the same, then we should get the biowaiver for this. And so just to show you that approach definitely, how can they justify the use of Weibull function in that case? I don't know who answered this question. I didn't raise it, but I'm just remembering that case. Definitely, one would not rely on just comparing the fitted the key parameters coming from the Weibull function to say if they are comparable. Then that's the end of story. You should give us the wider dissolution profiles or waive this. So that's sort of things that in that extreme case, there is no way that they can justify the use of a Weibull function to make a regulatory decision, right?
I would agree with that you should not be comparing just the parameters of the Weibull function, right? use
Right.
those parameters of the viable function to actually calculate what dissolution profile would be resulting from each of those viable functions and compare those profiles, not just the parameters.
Exactly, Vera. So that's one thing, right? Like, it's not justification in that way. And so usually, as Vera mentioned, and we know this for immediate release formulation, the Weibull function is not the best recommended, but for extended release formulations, we have widely used for years Weibull function to do even non-conventional approaches. And so I would say that experience is there and we need more justification. One, I would wonder whether they could use the Weibull function for an extended release formulation to extrapolate, let's say, to 100% when the initial data, the lab data, they didn't have that, they didn't generate that. So, we will be really looking into the impact of that extrapolated data for an extended release formulation. So hopefully that helps a little bit.
Yes, very helpful. Now, looking at the clock, I think we have another poll question, Arlene.
Yes, we do. Which of the following statements is or are true for PBBM-based mechanistic IVIVR? If you want to take a few moments to submit your answer, this roundtable discussion is also meant to further educate people who may not be GastroPlus advanced users or people who have done these IVIVC and IVIVR submissions for regulatory application. All right, we're going to go ahead and close the poll now. And there's the answer. Tycho, right back to you.
Great, thank you. And perhaps we can just use the few remaining minutes here for final closing thoughts from Sandra and whoever wants to contribute to from the panel members. Sandra, any final thoughts for this particular session?
I would say that safest space, the concept of safest space is growing, and pharmaceutical companies should take advantage of it, because I do believe that will expand the regulatory flexibility and will help you to accelerate your drug product development and hopefully have better drug products with patient-centric drug product quality in mind.
Thank you. Anyone else with final thoughts?
Just a quick final reminder that I'm sure that there is a lot more questions that these discussions generated, right, to the audience. So the GastroPlus user group LinkedIn page is a great discussion forum. So I would encourage everybody who has additional questions or questions we didn't get to just post them there and get the answers and discussion from your peers.
Thank you. Anyone else? If not, then I would like to thank the panelists and everyone who joined us today. And as Vera mentioned, please make use of the GastroPlus LinkedIn webpage. And as always, the recorded webinars will be available for users to reuse. And you will also see additional links posted on our GastroPlus LinkedIn webpage. With this, I hope that all of you will continue to use PBBM in your regulatory applications and also publish, so we can influence model-informed drug development. And with this, I'll give it back to Arlene.
Thank you, Tycho. Thank you, everyone. This concludes our webinar for today. Have a great day.